Our next presentation is Exo Biologics, and I'd like to welcome Romain Desraville, Chief Business Officer, to the stage.
Thank you for the introduction, and good morning, everyone. I'd like to thank the ARM organization for giving me the opportunity to present Exo Biologics today. So who are we? We are a clinical stage company focused on exosomes. We are one of the leaders in the field of exosomes, and we are based in Belgium. We are at the moment running a clinical trial, a phase 1/2 for a rare pediatric disease, which is called bronchopulmonary dysplasia. And we also have opened our manufacturing platform for collaboration with cell and gene companies in the world through our subsidiary called Exo Expert. So to start with, what are exosomes? So you probably know that most of the cells do secrete small vesicles, which we call extracellular vesicles. A subpopulation of them are exosomes. They are filled with lipid, proteins, and DNA, and they play a really critical role in cell-to-cell communication. More specifically, when you have exosomes from MSCs, they do play a role in immunomodulation and regeneration of the tissues. This is what you have here on the top right, and the ability to produce naive exosomes give you the opportunity to create therapeutics for this kind of indication related to inflammatory or regenerative, the regenerative space. But in addition to that, if you do have the technology to load the exosomes or the EVs, the extracellular vesicles, with a payload, then you can enter the field of cell and gene therapy by loading siRNA, mRNA, or DNA compound in the lumen of the EVs. And one of the interests of this approach is that the EVs are perfectly safe, they are totally natural, and so they will not trigger any immunogenicity response, and they will allow you also to do redosing. You can also load the EVs with a small molecule as well. But nonetheless, there is some challenges with within the EV field, and one of them is the ability to manufacture them in the GMP way. So, as you can see, there is a handful number of companies in clinical trials at the moment in the US and EU, and one of the challenges, as I mentioned, is the ability to move to an industrial manufacturing stage. Most of the companies are using 2D scale processes, which is easy to start a clinical trial with, but then it becomes very complex financially, but also to keep consistency in the way you do manufacture exosomes. One of the challenges is the fact that the exosomes is a kind of heterogeneous population, and it's very hard to always produce the same population. You have inter and intra variability because of the donors, because the exosomes are usually produced by MSCs, and you have to organize a consistent supply. And in addition, the fact that EVs is a relatively new field, there is no clear regulatory pathway to go to clinical and even to go to furthermore to commercial. So it's not easy for new companies to know what is the strategies they need to deploy to get to clinical. And so what we've achieved at Exo Biologics is to build a platform called Exopulse, which allows us to manufacture EVs in a large-scale way and in a very consistent way. So in our clinical trial, we produce our EVs by MSC that are obtained from umbilical cord tissue. There are several advantages, but the platform is also flexible in a way that we can use MSCs coming from adipose tissue or bone marrow. We are using a bioreactor-based process, which allows us to have different scale, which is very important for us in order to control the cost of the supply and also to adapt the amount of vesicles we need for each batches. So we do have a small scale, we do have an intermediate scale, and we are now working on an automation version of the platform. One of the key of the platform is the ability to increase the productivity of the cells and the number of vesicles that are produced by cells, which allows us to keep the manufacturing scale relatively small. We're not talking about hundreds of liters of bioreactor. It's a relatively small scale because we really bet on the productivity aspect. We've developed the purification technique, the characterization, which is extremely important as it is a new field. The regulatory body is very sensitive to the way we do characterize and understand the mechanism of action. And in Europe, as we are the first company to start clinical trials with MSC-derived EVs, we're actually also building the regulatory pathway with EMA and our reference body, the Belgian authority. And so our lead compound called Exo 001 is used in the context of the rare pediatric disease called bronchopulmonary dysplasia. So when with preterm babies, the lungs are not fully formed. So the standard of care is actually to have an intratracheal injection of concentrated oxygen. The challenge with that is that it triggers inflammation of the lung tissue, and then it can lead to issue in the development of the lung, of the brain, and of the baby in general. So it's related to inflammation, fibrosis, and other elements. What we do is that we use our EVs in the same route of administration, so in intratracheal route of administration, in order to reduce inflammation of the lung. On the left-hand side, you can see in mouse model a normal lung tissue. In the middle, you can see our model, which is replicating the bronchopulmonary disease. You can see that the tissue is damaged and you have less alveoli. And then on the right-hand side, you can see the model, but with the addition of our exosomes, and you can see that the lung tissue is similar to the healthy model. So this is one of the data we've submitted in our IMPD to show the anti-inflammation effect of our EVs. So we are currently running our phase 1 in Europe. It's an ODD. We're also talking with FDA to move to the US. One of the interesting aspects of the EVs and the exosome is that the approach is quite generous. It's a general anti-inflammation system. So with the same product, we can actually work with other indications without having to do a modification of the product. So it opens the gate to dermatology or gastroenterology or any other indications, and this is what we are focused on. So there is a small typo. We're not yet in phase 1 with dermatology, but this is our next focus with our compounds. So as I mentioned, the route of administration at the moment with the preterm babies is intratracheal. We know we can go IV, but we also wanted to demonstrate the possibility to work on nebulization, as it would open the gate for us to COPD or asthma or any other respiratory disease. So we worked on different systems in order to show if the nebulization would impact the effect of our EVs. And so on the graph on the right-hand side, this is our typical macrophage-based assay. We're looking at inflammation markers. The first bar is showing the inflammation, so we do have an inducer of the inflammation. This is a baseline level. The second bar is our negative control, and if you look at the two last bars, the two green ones, so the first one is just the EV alone without nebulization, and you can see that the inflammation level is reduced, which is expected, as this is what we're doing at the moment. And the last bar is a nebulization. And so you can see that not only we still have an inflammation effect, but we even have a further enhanced anti-inflammation effect. And so we're working in understanding why we have a stronger effect, but the good news is that the anti-inflammation effect is kept. So this was the first step to show that we can manufacture GMP-compliant EVs, and they are safe because we already closed the first cohort of preterm babies. And so we've been working on drug loading and delivery with our EVs. There are several advantages to that. The first one, obviously, is for those which are looking at alternative to LNP or AAV. One of the main elements being that the EVs are non-immunogenic, so you don't trigger any immune response. It's as it is natural, and you can also have redosing of the EV. And so it's exactly the same platform, but you can just see in green here the addition of a unit operation, which is the loading. So we work on EV post-production loading, meaning that we first produce the EVs and then we load them with the payload. We've worked on loading small molecule and siRNA, and at the moment we're also working at DNA loading and bigger RNA loading. And all of that is GMP compliant as well. So we don't need to have extra development to go to clinical if we have something working in animal. And so the first step was to load small molecule. Again, it's just on the left side, this is just showing the fact that we are loading our EXO-1 compound with small molecule. It's the same kind of assay, so inflammation-based assay. And if you look at the two last bar, you can see that we have, not the last one, but just before, this is the EV alone, which again is showing anti-inflammation. And then when we load with the small molecule, you see that we have an anti-inflammation effect. And the molecule itself is a corticosteroid. And so we can demonstrate that the effect is kept and is working in vitro. But the very interesting part for cell and gene therapy is the loading of siRNA. So again, looking at the same system and loading methodology, we worked at loading our EVs with siRNA and checking if the delivery was working. Again, it's the same anti-inflammation assay. And if you look at the right bar, so the first we tried to load or to compare the siRNA loading with LNP, just to see if we have a similar or better effect in vitro. And if you look at the two last bar, again, we have so first the EV, then the EV and the siRNA, but just co-incubated, and then the EV and the siRNA loaded. And again, you can see that we can induce the anti-inflammation effect, and we can reduce the number of protein related to this siRNA. And it's very important because although a lot of companies are able to load siRNA in EVs, it's another step to show that you can actually deliver the EV into the targeted cells and demonstrate the impact on the protein levels. So as I mentioned at the very beginning, we've created a CDMO called Exo Experts, so we can have collaboration with other cell and gene company and offering this non-viral drug delivery system, which is fully GMP and fully automated. I mean, we're working on the automation and it's going to be finished in 2025. And we are not siRNA or DNA experts, so this is why we want to collaborate with other companies that are looking for alternative way of delivering their payload. And this is just a last slide showing the few companies we're working with. For the moment, all of that is preclinical, and normally next year we will be able to start animal study with an siRNA compound that would be loaded in EVs. And if we're looking at partnership with any other company that have a payload and is looking into alternative towards the delivery as opposed to AAV or LNP. So feel free to reach out if you are interested. Thank you very much for your attention.